What we really want to understand about vaccines is how they work and how they work across the lifespan. We don't fully understand precisely what happens to our immune system as we get older. So there are lots of different theories, but really if we want to understand that, then we need to look at how our tissues are actually responding, what they're doing in real time, and that's what we're trying to do with this study. We're comparing responses in younger people and in older people. We ask people to come forward if they're interested in taking part, and we've been completely inundated. We had over 700 research participants saying that they would like to take part in our study. They'll be asked some questions about whether or not they can take part in the study, about medical history, what medications they might be taking, to make sure that it's safe to take part. Then if they've got through that stage, then we'll ask people to come forward and have some vaccines. In this study, we're using a flu vaccine and a COVID vaccine. And what we're doing specifically that's unusual is we're looking at the tissue that's responding at lymph nodes. Lymph nodes are small bean-shaped organs. We have them throughout our body, and they're a very important part of the immune system. They are important in taking small parts of foreign bodies, so germs, and then presenting those to other cells who then make a response to it. If we have a vaccine given as an injection in the arm, we get a response in the lymph nodes in the armpit, and that's where the cells and the antibodies that are going to protect us from infection are made in response to the vaccine. So if we really want to know how the vaccine is working, we need to look at the lymph nodes that are responding to see what they're doing and how they're making these cells and these antibodies so that we get the best response from our vaccine. In a series of visits that happen afterwards, we take lymph node cells using ultrasound-guided biopsy. You can think of it a little bit like a white blood cell test. So we're not taking cells from a vein, we're taking them actually from lymph nodes. So in order to do that, we need to be able to see them, and we can do that using ultrasound, and then we can use that to guide a needle and take a few cells. But then we're able to look at those cells in great detail in the lab. In some experiments, we look at them just one by one to really understand how they're responding. The whole process is relatively short. We follow people up for weeks rather than months or years to see how they're responding over time. And then we repeat in the next season with a new set of volunteers. Our participants are absolutely part of our team. We're really grateful to them for all the time they give, the support. We have a questionnaire where we're asking people about their experience with our research to make sure that the participant journey through research is the best it can be. And we really value that feedback from participants. This research programme is currently funded by the Chan Zuckerberg Initiative and the MRC UKRI. What I was keen to do was set it up as a programme so that we could bring in this multidisciplinary team. So we have radiologists, we have single-cell technologists, bioinformaticians, clinical research nurses, and that's such an exciting space to work in. And then we also have these fantastic laboratory facilities. We have a big team of people who can then look at those lymph node cells that we're taking and investigate them really in great detail. We can look at the genetics, we can look at the different proteins that are being expressed, and bringing those different components together then gives us this incredible detail for understanding how vaccines work. I believe that this research can have so much benefit. In this particular study, we're asking what happens as we get older. But we can ask lots of other questions using this approach. We've heard a lot about mRNA vaccines, for example, and really starting to probe how those are working in these tissues could be very beneficial for making the next generation of mRNA vaccines. My dream for this kind of research is that it gives us new insights that help us to design vaccines with a rational and novel approach